This is 7th period review. Say hi.
Hi.
And this is Honors Bio, and we are reviewing chapter 3, which is all about the cell. Okay, and the first thing we need to know is the cell theory. So we remember the parts of the cell theory? All living things are composed of cells. What's the second part? Functional unit of all life is the cell. And number 3, cells only come from other cells. Okay, so you got to remember that. Then the other big thing we need to know is that cells need to have a large surface area per volume. That's why cells are very, very, very what? Small. Because everything going in and out of the cell is going to cross that cell membrane. Okay, and we gave the analogy as if somebody sprayed perfume, if this were a cell and somebody sprayed it right here at the door, how many people would smell it versus if I sprayed it inside this little office versus if I sprayed it in the gym, right? Okay, so when you look at the cell now, we know we have a large surface area per volume. When you look at all these things on this picture, you should know what everything is and what everything does. Do you? Okay, good. Now, we started in talking about just prokaryotic cells. Prokaryotic cells, bigger or smaller? Smaller. Organelles or no organelles? No organelles. DNA or no DNA? They have DNA, but it's not enclosed within a what? Nucleus. It's just called the what where it hangs out? Nucleoid region. The nucleoid region is where it hangs out. Does it have ribosomes? Yes. What happens on ribosomes? Protein synthesis. Good. Okay. Does it have membranes? Yes. It just doesn't have membrane enclosed organelles. There are 3 layers going from the inside to the outside. We looked at it right here. Okay. What's this innermost layer? Cell. It's on every cell. That's the first layer you have. What is it? Cell membrane. What does the cell membrane decide? What goes in and out? What's the next layer out? A cell wall. And then what's the 3rd layer out? Capsule. It could be slimy or hard. Right? Those are the layers in a prokaryotic cell. Then we looked at hairy corn dog. Remember this? Okay, what's the blue? Cell membrane. The red is wall, and then the capsule. Then all these hairs, there was a name for them. They hang on. They're not really hairs, but they're called what? Starts with an F. Fimbriae. So it's for attachment. Some of these, you have little cytoplasmic extensions called a sax pilus, and you use that to exchange DNA. We already learned nucleoid region. We already learned, what else did we learn? Ribosomes for protein synthesis. Cell walls protection. What's the flagella for? Movement. Good. Okay. Ask your bio buddy, do you have any shadows casted on the prokaryotic cell? Things you don't understand that you need to shine some light on? Some light of wisdom? Flashlight of wisdom? Go ahead. Ask them. Do you know what a plasmid is? Do you know what a plasmid is? It's bonus DNA. Now, DNA is just like, welcome. This is a tutor for you to use at any time. If you would like extra help in biology, we will always have tutors here on Mondays and Tuesdays, 7th period. Student tutors that have done well in my class. Okay, so you can always work with them. Plasmids, oh, is it still recording? Yes. Plasmids are extra rings of DNA that can code for proteins. And genetic engineers like to manipulate plasmids so they can get a protein product of their choosing. If I was going to pick a typical bacterium to be my mascot, I would pick cyanobacteria because they do this. What's this? Nitrogen fixation, and they did the oxygen revolution. Okay? So they're really important. They're autotrophs. They're at the bottom of the food chain. All right, now make sure you can differentiate between the 3 domains. Why don't you just get next to somebody you're with right now and see if you can come up. Why don't we have the darkest shirted wearing person try to describe the 3 domains. Go ahead. Yeah, in your own words. So 2 of the domains are very similar. What 2 domains are very, very similar? Archaea and Bacteria. What's similar about them? Yeah, they're both prokaryotes. Okay, what's the difference between Archaea and Bacteria? Which one is, a little more on the edge, a little more extreme? Archaea. Yeah, they live where it could be really salty or where it could be really hot or it could have a really low pH. Which one evolved first, Archaea or Bacteria? Bacteria. Remember, Archaea means ancient, but it's misnamed. It's when they didn't understand. Okay, so you have Archaea and Bacteria. Eukarya, this is where you have the 4 kingdoms, we talked about. Plants, what would you want to say about plants? Remember that. They are all what? Autotrophs. What could you say about Protista? Usually what? Unicellular, yeah. Eukaryotic cells, but they can also be multicellular. What could you say about Animalia to set them apart? They're not autotrophs, they are heterotrophs. But who else is a heterotroph up there? Fungi. What kind? How are they different? Yeah, these are absorptive and these are ingestive heterotrophs. All right. Now, we talked about prokaryotes and eukaryotes. This is not drawn to scale. But the question has been is how could a eukaryotic cell have evolved? And you remember we talked about, did I talk about it in here? No, sorry. I'm going into a different class. Never mind. All right. Hopefully in your notes, I asked you to get a picture, labeled and an unlabeled diagram that you could use as reference. And you can find one that are labeled and then a little summary statement about each one. And the reason why I told you to get an unlabeled one is you could go and you could print it out and you could annotate it or use a pen or something and test yourself on it because that's what you're going to want to do. And know the structures, be able to recognize it. I could put a picture of an organelle up there, or I could put a description of a function, and then I would expect you to know what it does. So on a cell membrane, what would you say up here? What would be really important to know about the cell membrane? Say it louder. In and out, right. I would say it decides what goes in and it goes out. What's the key thing about the structure of a cell membrane? What's it made out of? Yeah, phospholipid bilayer. Good job. All right. Good. If we went on to another one, Golgi Complex. What does the Golgi body do? What's our analogy? UPS man, right? He's packaging. He's transporting. He puts on the address label of where something needs to go. Who brought the Golgi Complex something? Yeah, the endoplasmic reticulum, okay, ships it off to the Golgi, then the Golgi ships it off somewhere else. The ER, there are 2 kinds of ER. What are the 2 kinds of ER? Rough and what else? Smooth. Good job. Okay, the rough is rough because it has what on it? Ribosomes, and that's where proteins are synthesized. Okay, do not tell me on a quiz question that proteins are synthesized on the ER. You're going to tell me proteins are synthesized on ribosomes. Ribosomes could be on the ER, or they could be free floaters too. Okay, and you get fats in smoothie are. Fats and lipids and phospholipids. What's the nucleus? What would we consider that to be? The brain of the cell. It has DNA and it has proteins in it called chromatin. What about this guy right here? What is he? And what does he do? Powerhouse, right? What's the reaction that he does? Cellular respiration. Good job. Okay, peroxisome. What does a peroxisome do? It detoxifies, but it breaks down hydrogen peroxide. It breaks down that metabolic waste. Why am I? Oh, here, lysosome. What does a lysosome do? Stomach? Good. Stomach of the cell. What about these? What are these called right there? Centrioles. What do they look like? Churros. What do they do? Nobody knows. Okay. The function is unknown, but they sit in what's called a microtubule organizing center, or centrosome, that kind of anchors microtubules during cell division. But we still want to know where they are. Let's see. What kind of holds the structure of the cell together, just like the structure in us? Cytoskeleton. Okay, and we talked about there were three. There was the actin, those were the smallest ones of all, very fine. And then the largest ones of all were the microtubules. And remember how we watched it and it was like it would walk on the microtubules? Remember that? The cellular railway. Okay, good. You want to take some practice questions over this now and see how you do? Oh, I knew I talked about this over here. Okay, wait, I'm not ready for you to do that yet. This is a good diagram, how it shows the link between the nucleus, the ER, to the Golgi, to vesicles that you're going to make. Okay, let's look at this real quick. What's different? What's this for? Yeah, it has water in it, and it pushes, it keeps the cell upright, because it's like a vacuole is like the multi-purpose organelle, and it can be filled with water and push against the cell walls to keep it upright. It can be a contractile vacuole and protista to help with water regulation. It can be a storage vacuole. What's this for right here? What is that? Yeah, chloroplasts for photosynthesis. And what's this thing right here? Cell wall. What is it made out of? Remember we learned this with the 4 important organic molecules made out of cellulose? What doesn't the plant have? Yeah, there's no centrioles in plants. They don't have that. Do plants have mitochondria? Yeah, they still do cellular respiration, right? Right. So they still have mitochondria. Okay? These two are connected. Why? What do both of these cells, these organelles have? They have their own what? DNA. Remember, and it's in a shape like a circle, like prokaryotic cells. What else do they have? Their own ribosomes. They have a double membrane. And they're self-replicating. Do you remember when I talked to you about the endosymbiotic hypothesis, how eukaryotic cells, how the evolution of eukaryotic cells, that one organelle came to live, one prokaryotic cell went to live in another prokaryotic cell and became its organelle? That's the endosymbiotic, you can do that, endosymbiotic hypothesis. Okay, ask the person you're sitting next to, is there any stuff that you're not feeling comfortable with right now? I bet you can find online flashcards to test yourself on these. I will post the quizzes. Somebody told me I didn't put the quizzes up. Did they put that? Gave me a post-it. Okay, I'll put the quizzes up. Okay, one last thing. Transport, cilia and flagella. I think I talked to you about in cilia and flagella, it's a 9 plus 2. There's 9 doublets around and 2 in the center, whereas centrioles are 9 groups of 3. Yeah. And basal bodies, which anchor cilia and flagella, are the 9 groups of 3. All right, let's see how you do. What else do I have there? Then we finish this. I might have a practice one you can do too, a real one. Maybe. Go in as whatever you like. Your favorite breakfast drink. Lemonade. Chai tea. Chai tea latte. Yes, I like that. Are you all in? Who wants to be in? Put what? Wine trunk.
No, wine shrub.
And I'm going to pause you. Okay, let's check our answers. Powerhouse of the cell, mitochondria. I wouldn't say the chloroplast is the powerhouse. It's energy related and it makes sugars. But what makes you things that a cell needs to spin for its reactions is the mighty mitochondria. Number 2, what goes in and out is a cell membrane. Good job. Okay, I still did recording. Processed packages. You guys are doing awesome. Stomach, lysosome. I'm so proud of what's going on here. Cell wall is about protecting and supporting. I would not say the cell membrane is about protecting. The cell membrane's primary goal is to let things go what? In and out. Determines what goes in and out. I would also say vacuoles are not about protecting. Okay? The other things, yes, but not about protecting. Control center, nucleus, not the mitochondria. Why would I say the nucleus is the control center? It has the DNA. Yay. Meh. Okay, chloroplasts do photosynthesis, not mitochondria. Stores, transports, turgor pressure in plants, that is a vacuole. Site of protein synthesis, ribosomes. Okay, see, somebody put ER, right? We're not going to put that. Ribosomes can do protein synthesis and not be on the ER at all, so we wouldn't pick that. Plant and animal cells, mitochondria. Chloroplasts are not found in animal cells, right? Because what don't animal cells do? Photosynthesis. 9 sets of 3, centrioles and basal bodies. Good job for those of you that got that. 9 sets of 2, cilia and flagella. And that is it. Okay, so know your structures, know your cell theory, right? Know your functions, all of that, and you should be set. Okay, do you want to, I'm going to see if I have a practice one. Okay. And I will put up the quizzes. All right, have a piece of toast if you're hungry. Make good choices. Love you.